Okay, in the second year part, so the next lesson is the molecular basis of inheritance. Okay, in this let us discuss some of the important questions and the concept oriented questions which are expected in MCET exams. So let us come to the questions. Question number 1. The classical period of molecular biology began in 1953 with options 1. The discovery of DNA by Frederick Miescher. 2. The discovery of molecular structure of DNA, that is double helical model of DNA by Watson and Crick. 3. The discovery of transforming principle, that is experiment genetic material by Griffith and his co-workers. 4. Confirmation of genetic nature of DNA by Hershey and Chase. So here in the question it is related to the year 1953. So year 1953 is considered to be the, okay, from here onwards their the classical period of molecular biology have been started. Why? Because from here onwards, that means this is a year where the structure of DNA was deciphered by the Watson and Crick scientists. So here the DNA structure is called as a double helical structure. So this double helical structure was proposed by two scientists, Crick and Watson, with the help of the X-ray crystallography that was developed by Wilkins scientists. So likewise this is a year from where the classical period for molecular biology have been started. So question number 1, the correct answer is 2. Question number 2. A double-stranded DNA of E. coli in which both the strands are labeled with a heavy isotope N15 was transferred into a medium with NH4Cl in which nitrogen is a radioisotope of 14. This DNA undergoes replication per every 20 minutes. After 80 minutes, what is the percentage of DNA with N14, NH4Cl and N15 respective strands? Options 1. 50% and 50%, 2. 75% and 25%, 3. 93.73% and 6.25%, 4. 87.5% and 12.5%. Okay, here it is related to the double-stranded DNA that is present in E. coli. We know that E. coli is the one which is replicating for every 20 minutes, or sorry, it is dividing for every 20 minutes by a process called as binary fission. And here the DNA which is present in that molecule is of N15. That is the two strands of DNA are of N15 type. Then this one is kept in the medium of N14 and it is made to divide for 80 minutes. That is for every 20 minutes it is dividing, means it is dividing for 4 times. So now let us take this one having kept in NH4Cl. In the first generation the two strands will separate out. These two are the 15 strands. When they replicate in N14 medium, they will synthesize a new strand in which the nitrogen base contains N14. This also synthesizes a new strand where the strand contains N14. That means after 20 minutes, two DNAs are formed, one with 14 strand, one with 15 strand. Then after 40 minutes, the two strands are separated out. That is, here it is 15 strand and here it is 14 strand. These two strands also separate out. In that also one is 14 strand, one is 15 strand. Then after that here, as are kept in a 14 medium, here they start synthesizing a new strand which is of 14 type, and this also synthesizes a new strand which is also of 14 type. Here it also synthesizes a new strand which is also 14 type, and here it also synthesizes a new strand which is of 14 type. That means after 40 minutes we get total of 14, we get a total of 4 DNA molecules, in which one is a hybrid with 14 and 15, and this is also a hybrid with 14 and 15, whereas the remaining two have two strands which are 14 in their nature. Then let's come to the 60 minutes one. So in this 15 and 14 are there, 14 and N14, N14 and N14, N15 and N14. So now in the 60th minute, after 60 minutes, what happens is once again the replication will take place. These two strands will separate out, and these two strands also get separate out from this one. So from here it is 2, from here these two, from here the two strands will separate out, and from here also the two strands will separate out. Now each of them, so here it is already 15 strand, 14 strand, 14 strand, 14, 14, 14, this is 14 and this is 15 strand. Now this will synthesize a new strand which is a 14. This also synthesize a new strand which is a 14. This also synthesize a new one which is a 14. So each of them is synthesizing a new strand which is a 14 type only, because they are kept in a N14 medium. Like this. Now after 60 minutes we got a total of 8 DNA molecules, and now in 80 minutes what happened, the two strands will separate out, and already is one 14 and one is a 15 one. This will synthesize a new strand which is 14. This also synthesize a new strand which is 14. From this two strands are separated out, and already they are 14 and 14. They synthesize a new strand which is 14. This also synthesize a new strand which is 14. From this also two strands will separate out. Already they are 14 type. Then they also synthesize a new strands which are 14. Now let us see for this one. The same thing is for this one also. Already 14 and 14. They synthesize new strands which are of 14 type. Then for this one, the same process, 14 and 14 are there. So this, they synthesize a new one which is also 14 type. For this one, so let's write for this one here. So 14 and 14. Here also already it is 14 and synthesize one more 14. For this one also the same thing. Two strands are separated out. They will synthesize a new one 14 and 14. This also synthesizing the 14 and 14. For this one, let us write here. The 14 strand is there and one is a 15 strand, because one is 14 strand, one is 15 strand. So it will synthesize a new strand which is 14 type, and this is synthesizing a new strand which is a 14 type. So after 80 minutes we are getting a total of 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15 and 16 DNAs we are getting. So this 16 DNAs is equals to 100%. This 16 DNAs is equals to 100%. So here after 80 minutes, what is the percentage of DNA with NH4Cl and N15 respectively in their strands? So out of them you can see that this is the one with 14 and 15. This is the one with 14 and 15. Right? So all the remaining will be of the 14 and 14 strands. So how many strands are there in total? In total we have 32 strands. So 32 strands is equals to 16%. Then out of them how many are there with 14 strands, means out of that 100%, 93.73% are of N14 and 6.25% are of N15. So from the given options, question number 2, the correct answer is 3. Question number 3. The individual functional monomers of nucleic acids are options: nucleosides, nitrogen bases, nucleotides and pentose monosaccharides. Okay, here what is a nucleic acid? Nucleic acid is an polymer of nucleotides. That is, many nucleotides are present in the form of a long chain-like structure called as a polynucleotide strand. So that from the given options we say that nucleosides does not include a phosphorus group in them. Nitrogen base is one of the part of the nucleotides. So pentose monosaccharides are also the part of nucleotides. So when we are saying it as the functional unit of nucleic acid is nucleotide. So question number 3, the correct answer is 3. Question number 4. The number of phosphodiester bonds present in pi into 174 bacteriophage DNA molecule. Options: 1. 5386, 2. 48502, 3. 5385, 4. 5384. Okay, here this is related to the number of phosphodiester bonds present in the nucleic acid of pi into 174 bacteriophage. In this bacteriophage, it is related to the number of nucleotides. So they have 53,086 nucleotides, and the phosphodiester bonds are one less than that one, that is 5385. So question number 4, the correct answer is 3. Question number 5. The number of nucleotides present in the double stranded DNA of Escherichia coli. Options: 97,004 base pairs. Second, 3.3 into 10 to the power of 9 base pairs. 3. 4.6 into 10 to the power of 6 base pairs. 4. 5386 base pairs. Okay, here in the textbook it is directly given as the nucleotides which are present in them is 4.6 into 10 to the power of 6 base pairs. That is the number of base pairs present in that Escherichia coli bacteria. So question number 5, the correct answer is 3. Question number 6. The X-ray diffraction data for DNA was produced by. Options: 1. Watson and Crick, 2. Maris Wilkins and Rosalind Franklin, 3. Frederick Michaud, and 4. Altman. So here we know that double helical model was proposed by Watson and Crick, but the person who helped them in studying that structure is Wilkins and Franklin, because they had gone for X-ray diffraction data, which helped Watson and Crick for proposing the double helical model. So X-ray diffraction for DNA was produced by Wilkins and Franklin scientists. So question number 6, the correct answer is 2. Question number 7. In double stranded DNA, the A plus G by T plus C is equals to 1. Existence was first reported by. Options: 1. Wilkins and Franklin, 2. Watson and Crick, 3. Michaud and Franklin, 4. Chargaff. So here we have to know something about the relationship between the purines and pyrimidines which are present in a DNA molecule. That is the number of purines in one strand of DNA are equal to the number of pyrimidines in the other strand of same DNA molecule. Same DNA molecule. So that means purines number is equals to pyrimidines number. We know that purines are adenine and guanine. Pyrimidines are thymine and cytosine in DNA molecule. And here always purines are complementary to pyrimidines. That is wherever the adenine is coming on one strand of DNA, the thymine will be present on the opposite side of the opposite strand. Then when guanine is present on one strand of DNA, so cytosine will be present on opposite side of the second strand. So like if you take the diagram here, this is a complementary, we can say the coiling of the DNA molecule. in this, for example, here if A is coming here, so opposite to this one, thymine is present. The number of bonds present between will be 2. And wherever if G is coming here, the number of hydrogen bonds will be 3 and complementary to it is C will be present. And whenever thymine is coming, the opponent will be having A, the number of hydrogen bonds present between will be 2. The same way, wherever if cytosine is coming, guanine is present in the opposite strand with 3 hydrogen bonds. So likewise, the complementary nature will be there. That means whatever the number of adenine plus guanine, that is equals to thymine plus cytosine. That concept of ratio, that is proportion of purines and pyrimidines in 1:1 ratio, was given by a scientist called as Erwin Chargaff. So question number 7, the correct answer is 4. Question number 8, the unique property of polynucleotide chain is options: 1. Occurrence of base pairing, 2. Formation of phosphodiester bonds between nucleotides of opposite strands, 3. Formation of glycosidic bonds between base pairs, 4. Formation of hydrogen bonds between successive phosphates. Okay, here the question is related to the polynucleotide chain. The polynucleotide chain means it is related to nucleic acid. Poly, many, nucleotides. Many nucleotides are present in the form of a chain-like structure. So here what is the unique property of this one is the formation of phosphodiester bonds between nucleotides of opposite strands. It is not related to this one. Then formation of glycosidic bonds between base pairs. Actually here between the base pairs, like adenine is there, thymine is present, the type of bonds which are present between them are the hydrogen bonds, but it is given as a glycosidic bond in the third option, that is incorrect. Then fourth one, formation of hydrogen bonds between successive phosphates. So here hydrogen bonds are present between the what do you call this purines and pyrimidines, that is adenine, thymine and guanine and cytosine of opposite strands. So that is also incorrect. So second one, formation of phosphodiester bonds between nucleosides of opposite strands, that is also incorrect. So occurrence of base pairing, already we said that the purines are pairing with the pyrimidines, that is adenine is pairing with thymine with 2 hydrogen bonds, whereas guanine is pairing with cytosine with 3 hydrogen bonds. That occurrence of base pairing is one of the unique features of the polynucleotide strand called as a DNA molecule. So question number 8, the correct answer is 1. Question number 9, in one complete turn of double stranded DNA, theoretical ratio on the basis of randomness of the ratio of adenosine, guanosine, thymidine and cytidine respectively. Options: first one 2:1:2, 2:1. Second one 2:3:4:2. Third one 1:1:1:1. Fourth one 3:2:1:1. So here it is related to the nucleosides that have been given here. Then what is the ratio between the nucleosides? Whatever the ratio that is there between the nitrogen bases, that same type of ratio will be there theoretically, that is 1:1:1:1. So let us come to first adenosine. Adenosine is a nucleoside which is formed by combination of adenine plus the deoxyribose sugar, that is a pentose sugar in DNA molecule. And second one is guanosine. It is formed by guanine plus deoxyribose sugar. Then next one is thymidine. Thymidine is a nucleoside which is formed by thymine plus deoxyribose sugar. And cytidine. Cytidine is formed by cytosine plus deoxyribose sugar. As we know that theoretically we say that the purines and pyrimidines are equal in their proportion. That is they are existing in 1:1:1:1 in one complete turn of DNA, but they may be practically differing. So when we are saying all these are equal in their proportion, definitely the nucleates are also will be in equal ratio theoretically, that is they exist in 1:1:1:1 ratio. So question number 9, the correct answer is 3. Question number 10, the elements found in nucleic acids are options 1 carbon, hydrogen, oxygen, nitrogen, phosphorus and potassium. 2 carbon, hydrogen, oxygen, nitrogen, phosphorus. Third one carbon, hydrogen, oxygen, nitrogen, phosphorus and sulfur. 3 carbon, hydrogen, oxygen, phosphorus and potassium. Okay, here nucleic acids, we know that nucleic acids contain the units called as nucleotides. Nucleic acids are made up of units called as nucleotides, and each nucleotide contains a nitrogen base, a ribose sugar plus a phosphate group. Nitrogen base is a dicyclic compound or a monocyclic compound which is made up of carbon nitrogen rings. That means they contain carbon, nitrogen and somewhere they contain oxygen also along with hydrogen. That means the nitrogen base are made up of carbon, nitrogen, oxygen and hydrogen. Then coming to ribose sugar. The formula of ribose sugar, that is here the deoxyribose sugar is there, that is pentose sugar is present here, and or sometimes it is ribose sugar or deoxyribose sugar based on the RNA or DNA molecule. They contain carbon, hydrogen and oxygen. Ribose sugar is made up of carbon, that is pentose sugar is made up of carbon, hydrogen and oxygen. When it is coming to phosphoric acid, it is PO4, it contains phosphorus and oxygen. So from this we can say that carbon, hydrogen, oxygen, nitrogen and phosphorus, these are the 5 elements which are present in the nucleic acids. So from the given options, question number 10, the correct answer is 2. Question number 11, a normal DNA molecule is allowed to duplicate once on a medium containing radioactive thymidine. What will be the correct number of DNA strands that contain non-radioactive thymidine? Options 1, 4, 2, 8, 3, 2 and 4, 6. Okay, here we know that DNA can replicate. Replicate means the DNA will give rise to the daughter DNA molecules. Daughter DNA molecules. That is, if this is a DNA molecule, at one point it will the 2 strands of the DNA will separate out, and each of the strand is acting as a template, and each of the strand will synthesize a new strand like this, start synthesizing it, and these are called as the daughter DNAs. And in them, where does this replication is taking place or duplication is taking place means where the radioactive thymidine is present. Radioactive thymidine is present. Then what will be the correct number of DNA strands that contain non-radioactive thymidine? Radioactive thymidine is present in a new strand which is synthesized, this one which contains a radioactive thymidine here, and in this daughter DNA also this strand will contain radioactive thymidine, but each of the daughter DNA contains one old strand. This does not contain radioactive thymidine because it is already having the thymine in it, and in this daughter DNA also this strand does not have the radioactive thymidine because it is coming from the parent. So we know that according to semi-conservative method of replication, each daughter cell will conserve one strand of the parent, so that the number of strands which are not having radioactive thymidine is 2 strands, that is one in one of the daughter strand and one of the daughter DNA and one strand in the second daughter DNA. So question number 11, the correct answer is 3. Question number 12. DNA is absent in? Options: 1. Chloroplast, 2. Mitochondria, 3. Plasmids, and 4. Ribosomes. Okay, now let's come to the first one, the chloroplasts. Chloroplasts are the cell organelles which are present only in eukaryotes, and these chloroplasts are considered as semi-autonomous cell organelles due to presence of circular, naked, double-stranded DNA molecule in it. Okay. Then second one is mitochondria. Mitochondria is also present only in eukaryotes. This is also considered as a semi-autonomous cell organelle due to presence of a circular, naked, double-stranded DNA in it. Then coming to plasmids. What are plasmids? Plasmids are the extra chromosomal or extra nuclear DNA molecule present in bacteria like in Escherichia coli and Salmonella. So here this is nothing but the circular, naked, double-stranded DNA. So this is also then coming to ribosomes. Ribosomes are considered as the smallest cell organelles which are chemically called as nucleoproteins. Chemically called as nucleoproteins. Nucleo means they contain a nucleic acid and they contain the proteins also. What is a nucleic acid by which the ribosomes are made up of? These are made up of ribosomal RNA. That is rRNA is present, but there is no DNA in it. So ribosomes are the one which does not have DNA in it. Question number 12, the correct answer is 4. Question number 13. A typical double-stranded DNA molecule with a length of 1360 angstroms undergoing replication in medium containing deoxyribonucleotides. After 3 generations of replications, how many deoxyribonucleotides are consumed from the medium? Options: 6400, second 6000, third 4800, fourth 5600. Okay, here the question is related to a DNA molecule whose length is 1360 angstroms. We know that in general one coil have a length of 34 angstroms which contains 20 nitrogen bases in it. But in the question the length of the DNA is 1360 angstroms. So it is forming 40 coils. When 40 coils are there, each coil is having 20 nitrogen bases, 20 into 40. It is coming to 800 nitrogen bases in that DNA which is of 1360. If this DNA molecule is made to replicate for 3 generations in a medium containing deoxyribonucleotides, 3 times means here in the first generation we know that the DNA was having initially 800 nitrogen bases. After first generation the number of nitrogen bases, that is each strand contains 400 nitrogen bases. So here one strand with 400 and one strand with 400 are separated out. Then after first generation the second one more strand is synthesized which forms 400 nitrogen bases. Here also one more strand is produced which is having 400 nitrogen bases. That means after first generation how many deoxyribonucleotides are consumed? Here it is a total of 800 ribonucleotides are utilized. After in the second generation these 2 strands are separated out. These 2 strands are also separated out. Now each of them will synthesize a new strand. That means 400 ribonucleotides are taken from the medium. Here also 400 ribonucleotides are taken from the medium. Here also 400 ribonucleotides and here also 400 ribonucleotides. That means in the second generation the total number of ribonucleotides that are taken are 1600. Then when you see the third generation, let's continue this one here. Third generation, the 4 of them are separated out. Like from here, 2 are separated like this, and 2 are separated from this one. From this one also 2 are separated out, and from this one also the 2 strands are separated out. Then this one will synthesize a new strand where 400 ribonucleotides are taken. This also synthesizes a new strand which is taking 400 ribonucleotides. The same way for all the 4. So that every time they are taking 400 ribonucleotides when they are undergoing duplication. When they are undergoing duplication. So that in the third generation a total of 3200 ribonucleotides are taken from the medium. In the first generation it is 800, second generation 1600, third generation is 3200. So now if we calculate, add all of them, 3200 in the third generation, 1600 in the second generation, and 800 in the first generation. If we add all the ribonucleotides, we are getting 5600 ribonucleotides. That means when a DNA with 1360 angstroms is made to replicate in a medium for 3 generations where deoxyribonucleotides are there, they are consuming a total of 5600 ribonucleotides from the medium. So question number 13, the correct answer is 5600, that is option number 4. Question number 14, arrange the following in ascending order based on their molecular weight. Options: A. Nucleoside, B. Nucleotide, C. Nitrogen base, D. DNA of bacteriophage lambda. The given options are C, A, B, D. Second option B, A, C, D. Third option A, B, C, D. Fourth option C, A, B, D. Okay, here the 4 that have been given are related to the DNA molecule only. So in that one we can say that nitrogen base is the one which is having lowest molecular weight. Why? Because it is a part of nucleoside. So definitely the nucleoside will have more molecular weight than nitrogen base. Then what about the nucleotide? So here let us take here first one, nitrogen base with the least nucleotide, with the least molecular weight. So that is a C. Then it will be the A, nucleoside, because nucleoside includes nitrogen base plus pentose sugar. Then next one will be the nucleotide. That means nucleoside plus phosphate molecule. Nucleoside plus phosphate molecule. And the last one will be the DNA of bacteriophage, because DNA is a polynucleotide strand. That means each DNA contains such many types of nucleotides in it. So the correct order is C, A, B, D. That is the ascending order based upon the molecular weights. So question number 14, the correct answer is 1. Question number 15. The ratio between number of template strands which participate from a DNA molecule for replication and transcription process. Options: 1, 1:1, 2, 2:1, 3, 1:2, and 4, 2:0. Okay, here it is related to 2 processes that have been done by DNA molecule. One is replication. The second process is called as a transcription process. Replication is nothing but the doubling of the DNA molecule or duplication of DNA molecule. So when we talk about the replication process, this is a DNA molecule where the 2 strands are separated out, and each of the strand of DNA will act as a template. If it is 5' end, so this one will be the 3' end, 3' and the 5' end. So both the strands are acting as a templates, so that each of the strand will synthesize a new strand like this. So for replication, how many strands are acting as a templates? Means 2 strands of the DNA are acting as a templates. What about the transcription? Actually, transcription is the process of synthesis of RNA, that is mRNA from DNA molecule. When transcription is taking place with the help of the enzyme called as a polymerase enzyme, what happens is the 2 strands of DNA are separated at one place. This is a DNA polymerase enzyme. And the uncoiling of the 2 strands is a temporary process. Out of that, one is helping in the formation of mRNA. So out of the 2, we can say one is a sense strand, one is an anti-sense strand. The DNA which is helping in the formation of mRNA is called as a anti-sense strand. So based on that one, the mRNA synthesis will be taking place. For example, here on this DNA strand, if it is A is there, U will be formed, T is there, A is formed, G, here C will, likewise only one strand is helping in the formation of this mRNA molecule. So that here only one strand of DNA is acting as a template for formation of mRNA during transcription process. So the ratio between the strands of DNA which are acting as templates for replication and transcription is 2:1. Question number 15, the correct answer is 2. Question number 16, the cloverleaf model of tRNA contain options 3 accessory loops, 1 main loop and 2 tails. Option number 2, 1 accessory loop, 3 main loops and 2 tails. Third option, 1 accessory loop, 3 main loops and 1 tail. Fourth option, 2 accessory loops, 1 main loop and 1 tail. Okay, here if you see the structure of tRNA molecule exhibits a clover leaf model. Okay, in this one, if you see the structure of this tRNA molecule, we are having a clover leaf like structure like this. Okay, this is a one which is having lateral loops also. And this is the actual structure of a tRNA which is called as clover leaf model. So here, this is a site where the amino acid is binded. We call this as a tail end or amino acid binding site. And here 3 main loops are present, 2 lateral loops and 1 anticodon site. Here 3 unpaired nitrogen bases are present, that is called as a anticodon. So we call it as an anticodon end. Okay. Then it contains 1 accessory loop. 1 accessory loop. So out of the 2 lateral loops, lateral main loops, one of them is useful for identification of ribosome where the protein synthesis is taking place, and one more lateral loop is there which is useful for identification of the enzyme which will activate the amino acids. So that a total of 3 main loops are present. 3 main loops are present, 1 accessory loop is present, and 1 tail end is present. 1 tail end is present or 1 tail is present. So likewise in the given options we can say that 1 accessory loop, 3 main loops and 1 tail loop is a exact answer that is related to the clover leaf model of tRNA molecule. So question number 16, the correct answer is 3. Question number 17, among the following, which of the DNA molecule would have the highest number of hydrogen bonds? Options 1. Here 5' end to 3' end is given, where in one of the strand A and AA, and the second strand the complementary T's are given, 8 T's have been given from 3 to 5' end, and the second one 5' AAA TT AA is given to the 3' end, and in the second strand 3' TTT AA TT 5' end is given. Third one 5' AA GCG AGT 3 prime, and second strand have 3 prime TT CGC TCA 5 prime. The fourth option 5 prime GCAA TAA 3 prime, second strand 3 prime CG TTA TT 5 prime. So these are the options have been given where we have to find out the number of the strand with the highest number of hydrogen bonds in it. So let us take the first one, 5 prime to 3 prime, and second strand with 3 prime to 5 prime. In this, the total number of nucleotides that are given are 8. Adenine, 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 adenine. Total of 8 adenines have been given. So the complementary will be thymine, 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 like 8 thymines will be there. And between adenine and thymine, the number of hydrogen bonds are 2. Here also 2. Everywhere, wherever adenine and thymine are there, the number of hydrogen bonds will be 2. So as we say that 8 pairs are there, 8 into 2. The total number of hydrogen bonds that are present in the first option are 16 hydrogen bonds. Then coming to second one. Here the ratio is, the proportion is given as 5 prime, A, T, T, and A, 3 prime. The second strand T, 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 A, T, T, 5 prime. Wherever adenine and thymine is there, already I said that 2 hydrogen bonds are present. So everywhere we have to take 2 hydrogen bonds. But here how many pairs of A and T are there? Only 7 pairs are there. So 7 into 2, 14 hydrogen bonds are present for the second one. Now let us come to the third one, where 5 prime A, G, C, G, A, G, T, 3 prime is given. So the complementary strand will be T, T, C, G, C, T, C and A, 5 prime. Here A and T pairs are there, 2 bonds, but here G and C are there, 3 bonds. Here also 3 bonds. Wherever G and C are coming, we have to show with 3 bonds in it. Then here 2 bonds, 3 hydrogen bonds and 2 hydrogen bonds. So now if we count the total number, 1, 2, 3, 4. 4 A and T pairs are there. So 4 into 2 hydrogen bonds, that is 8. And GC pairs are 1, 2, 3, 4. So 4 into 3, 12 hydrogen bonds. A total of 20 hydrogen bonds are present in third one. First one with 16 hydrogen bonds. Second one with 14 hydrogen bonds. Third one with 20 hydrogen bonds. And let us come to the fourth one. 5 prime G, C, A, T, A and A, 3 prime. And here 3 prime complementary C, G, T, A, T and T, 5 prime. So if this is the case, we know that 3 hydrogen bonds here, 3 hydrogen bonds, 2, 2, 2, 2 and 2. So how many G and C pairs are there? 2 pairs are there. So 2 into 3, 6 hydrogen bonds, and here AT pairs are 5. So 5 into 2, 10. A total of 16 hydrogen bonds are present in the fourth one. So out of that, which one is having the highest number of hydrogen bonds means third one. That is based on the number of G and C pairs and A and T pairs. So question number 17, the correct answer is 3. Question number 18. A mRNA molecule has 66 nitrogen bases, including stop codon. What is the total number of functional codons and amino acids in a polypeptide chain respectively? Options 1, 22 and 21. Second option 22 and 23. Third option 21 and 21. Fourth option 22 and 22. Okay, here this is a question related to mRNA molecule. An mRNA molecule which contains 66 nitrogen bases. when 66 nitrogen bases are there, so when we say it's a triplet codon, it's a triplet codon means every 3 nitrogen bases will form into one codon. So that number of nitrogen bases by 3, we get a total of 22 triplet codons. And out of the 22 triplet codons, the last one is considered as stop codon. So that means we have to remove that stop codon from 22. So that out of that 21 are acting as a sense codons. What is the meaning of sense codons? Means these codons are specified for at least one amino acid. Somewhere they are specified to amino acids. So out of them, we can say that sense codons are nothing but the functional codons which are specific for amino acids. And here the question is related to functional codons and amino acids. As I said that all sense codons or all functional codons are specific to at least either of the amino acids. So that number of amino acids which are formed on protein chain when these 21 are present is 21 amino acids. So the answer is 21 and 21, that is 21 functional codons and 21 amino acids. So question number 18, the correct answer is 3. Question number 19, a DNA segment in which both the strands are labeled with N15 is transferred to N14 medium and allowed to replicate 3 times. How many daughter molecules show both strands with N14? Options 1, 4, 2, 8, 3, 6 and 4, 2. Okay, here the question is related to the N14 strands which are present in the DNA molecule. So there is a DNA molecule in which both the strands are of N15. So this DNA is kept in a medium where N14 is present. It's kept in a N14 medium and it is allowed to replicate. When it is replicating, the 2 strands are separated out. The 15 strand and the one more 15 strand is separated out, and this will synthesize a strand which is 14 because this is kept in a N14 medium. This also synthesizes a new strand which contains N14. Then this is the first replication. But the question is related to 3 times it have to be replicated. First generation, in the second generation, these 2 strands will separate out. That is a 15 strand is separated out and the 14 strand is separated out. So here they are synthesizing a new strands which are of 14 each. Here also the same thing. That is 2 strands are separated out, the 15 strand and the 14 strand. And each of the strand will synthesize a new strand which is of N14. This is after second generation. Then after third generation, the 2 strands are separated out here also. That is 15 strand and 14 strand here. They synthesize a new strand which is 14 and 14. Here the 2 strands are already 14. They will synthesize a new strands which are also 14 type. Here the 2 strands are already 14. They also synthesize a new strand which is of 14. And here the 2 strands are separated out. Each of them, out of the 2, one is having 14 strand, one is having 15 strand. So here both of them will synthesize a new strand which is of 14 type. Now after third generation, we get a total of 8 daughter DNA molecule. So the question is, how many daughter DNAs have both strands with N14? So this is a one which is having one N14 and one N15. So this is a one with both N14. This is also having both N14 here also, here and also here. When it is coming to this one, it have one N14, one and one N15. So that out of the total number of 8 DNA molecules that are formed after 3 generations, 6 DNA molecules are with both N14 strands. So question number 19, the correct answer is 3. Question number 20, with reference to DNA replication, which of the following statement is incorrect? Options: 1. DNA polymerase catalyzes polymerization of nucleotides in 5' to 3' direction only. Second, the replication begins at replication fork. Third, the leading strand is synthesized on 3 to 5' template strand. 4. The lagging strand also synthesizes on 3 to 5 strand, 3' to 5' strand after the synthesis of leading strand 5' to 3' direction. Okay, let us come to identify the incorrect statements from the options. So, we have to identify the incorrect statements. First one, so these statements are related to the DNA replication. So, we know that the DNA polymerase will catalyze the polymerization of the nucleotides, and always it will be in the 5' direction to 3' direction only. It never do in 3 to 5' direction. So, this statement that is given here, it is correct. The second one is replication begins at replication fork. Actually, when the DNA is undergoing replication, it is starting at a point. This point where the DNA is replication starting, what that is called as replication fork. So, this statement is also correct. Then the third one is the leading strand is synthesized on 3 to 5' template strand. So, like if the two strands are separated out, that is if it is a 5' direction, it is a 3' direction. So, the leading strand is synthesized on the this manner. So, likewise it is on the 3 to 5' direction on the template. Then this statement is also correct. And here it is coming to the last statement, the lagging strand also synthesized on 3 to 5 strand after synthesis of leading strand is incorrect. So, that fourth statement is incorrect. So, question number 20, the correct answer is 4. Question number 21, DNA replication generally takes place by options. 1. Semi-conservative and bidirectional method involves both by leading and lagging strand. 2. Conservative, bidirectional and only by leading strand method only. 3. Semi-conservative, bidirectional and lagging strand method only. 4. Conservative, unidirectional and leading strand method only. Okay, here this is also the question related to the DNA replication process. Here DNA replication means the doubling of DNA molecule. That is, a parent DNA will undergo replication to produce two daughter DNAs, and each daughter DNA will conserve one of the strand of parent DNA. That is, the daughter DNAs which are formed, they contain one old strand and one new strand. That is called as semi-conservative method. Then bidirectional. So, that is it is always taking place on two strands. So, it is bidirectional in their nature. That is 5', both of them it is from 5' to 3', but one is moving in this direction, the other will be moving in this direction. That is leading strand and the lagging strand are formed. So, here both leading strand method and the lagging strand methods are present here. So, as both the strands are present, so we can consider that all the four characters, that is a semi-conservative, bidirectional, leading strand method and the lagging strand, all the four are correct related to the DNA replication process. So, question number 21, the correct answer is 1. Question number 22, which of the following organisms does not follow the central dogma of molecular biology? Options. 1. Chlamydomonas, yeast, bacteria and HIV. Okay, here let's start the first one, Chlamydomonas. Chlamydomonas is a green alga, and this is a eukaryotic organism. When you are saying the eukaryotic organism, so here one more thing, that this is a one where the replication and also the translation, here the transcription and also translation are taking place. So, central dogma means the DNA is giving rise to mRNA molecule, which in turn helping the formation of protein molecule. here the transcription process is taking place, and here the translation process is taking place. In Chlamydomonas, DNA undergoes replication, DNA undergoes transcription to form mRNA, which in turn undergoes translation to produce proteins. So that is correct. And coming to the yeast, yeast is an unicellular fungi. This is also eukaryotic, and in this also the same process, that is DNA to mRNA by transcription, mRNA to protein by translation is taking place. And of course bacteria is an prokaryote. This also show the same character despite of the prokaryotic nature, DNA to mRNA, mRNA to protein by transcription and translation. When it is coming to HIV, human immunovirus, so here this is a one which is a virus which is acellular, and it does not show its metabolic activities by itself. So this is a one which does not show this type of central dogma of molecular biology. Question number 22, the correct answer is 4. Question number 23, arrange the following steps in sequential order with reference to the experimental proof of semi-conservative method of DNA replication in E. coli by Messelson and Stahl. Options: A. Culturing of E. coli bacteria in heavy isotope of N15H4Cl medium. B. Incorporation of heavy nitrogen into newly synthesized DNA and other molecules of E. coli. C. Transfer of cells into normal N14H4Cl medium. D. Separation of various samples on CSCl gradient to measure the densities of DNA of N14 and N15 types. Options: first one A, B, C, D, second one B, C, D, A, third one C, B, A, D, fourth one C, D, B and A. So it is related to the experiment of semi-conservative method of replication by Messelson and Stahl. So what he did is, he had taken the DNA molecule and or we can say he had taken the E. coli which is having the DNA molecule and kept in a heavy isotope of N15H4Cl medium. So first comes A. Then when it is kept in N15H4Cl medium, what happens is when the replication is taking place, the nitrogen in the form of heavy isotope, that is N15, is taken by the DNA and helping in the formation of replication process. That is, the new DNA which is synthesized contains N15. So the second one is B. Then transfer of the cells into normal N14 medium. So that is the next step, that is C. Whereas after that, the separation of these samples with N14 and N15 with cesium chloride. So that is a fourth step based on the densities. So the correct sequence is A, B, C and D. So question number 23, the correct answer is 1. Question number 24, the double-stranded DNA molecule labeled in both the strands with heavy isotope N15 is transferred to N14 medium and allowed to undergo replication for 3 generations. What is the percentage of DNA strands with N14 and those with N15 respectively? Options: 1. 87.5 and 12.5, 2. 12.5 and 87.5, 3. 50% and 50%, 4. 75% and 25%. Here the DNA which is having heavy isotope, both strands with N15 are there. 2 strands are with N15 and they have been kept in a medium in of N14 and they are allowed to replicate for 3 generations. We know that after 3 generations, the total number of DNA molecules that are formed is 8 DNA molecules. Out of the 8 DNA molecules, okay, 2 DNAs are with one N14 strand and one N15 strand. Whereas the 6 DNA molecules have both N14 strands. Both N14 strands. So the question is, what is the percentage of DNA strands with N14 and N15? Here the total number of DNA strands that we got is 16 DNA strands. 16 DNA strands, out of which here 6 DNAs are with both N14, that is 12 strands, and here the 2 DNAs with one of them with N14. So total 14 DNA strands are with N14. The remaining 2 DNA strands are with N15. So if we consider this 100%, this 16 strands as 100%, then the 14 strands are having this. The 14 strands are having N14, so they are accounting for 87.5%, and the 2 DNA strands which are having N15 are accounting for 12.5%. So question number 24, the correct answer is 1. Question number 25, the length of bDNA has 45,000 base pairs. How many complete turns will be DNA molecule take? Options: 1. 4,500, 2. 45,000, 3. 45, 4. 450. Okay. Here we know that one coil or one turn of DNA contains 20 nitrogen bases. 20 nitrogen bases, or it is 10 base pairs. Nitrogen base pairs are only 10 in one coil. So, but the question says that here 45,000 base pairs are present. 45,000 base pairs are present. How many turns does this bDNA will have? So, if we cross multiply them, 45,000 into 1 by 10 base pairs. So, if we strike off these zeros, it is coming to 4,500 turns. That is a bDNA which is having 45,000 base pairs will have a complete turns of 4,500. So, question number 25, the correct answer is 1. Question number 26. In eukaryotes, RNA polymerase 1 transcribes options: 1. 28S, 18S, 5.8S, 2. 23S, 5.0S, 16S, 3. 28S, 23S, 18S and 5.8S, 4. 28S, 16S, 18S and 5.8S. Okay, here in eukaryotes there is only one, in eukaryotes the polymerase enzyme is there which is helping in the transcription process. So, here during the transcription process it is transcribing 28S, 18S and 5.8S ribosomes. So, that here question number 26, the correct answer is 1. Question number 27. 3' AT GC AT GC AT GC AT GC 5' template strand, 5' TA CG TA CG TA CG TA CG 3' coding strand. Identify the sequence of RNA transcribed from the above DNA strands. Options: 1. 3' UACG 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 3'. Second one, 5' UACG 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 3'. Third option, 5' UAGC AUGC AUGC CAUGC 5'. Fourth one, 3' AUGC 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 5'. Okay, here in this the template strand, 3' template strand and 5' coding strand have been given. So, here template strand is 3', that is AT GC AT GC AT GC and ATGC to 5 prime end is given. So here when this is a coding strand means, so this is a antisense strand or the template strand of the DNA which help in the formation of mRNA synthesis. So here if this is a coding, this is a what you call the template strand. So the mRNA which is formed will be 5 prime end where adenine is present. So in place of adenine, thymine should come, but it is an mRNA molecule. Instead of thymine, it here uracil will come, and thymine, adenine. So complementary we have to write, where G, C, C, G. Here A is there, U will come. T, A, G, C, C, G. A is there, U. Here A, C, G. A is there, uracil will come. So here A, C and G, 3 prime. So this is a sequence of mRNA which have been formed on the template strand which is having this type of sequence. So from the given options we can say question number 27, the correct answer is 2.